Hi, I'm Christina Andreasen, and I'm an assistant professor at the Department of Pathology, University of Southern Denmark. My research focuses on how aging as well as pathological conditions such as Charcot foot and bone metastases affect the bone remodeling. And I'm also an editorial board member of HubLE, and today I'm actually here on behalf of HubLE to interview Ankita Duseja, who presented her work at ECTS Conference 2021. So, Ankita, could you please present yourself to the HubLE audience?
Hi, Christina. Thank you for having me over on this platform and giving me the opportunity to share my work. I'm Ankita Duseja, a PhD student over here at the University of Sheffield. And my project mainly focuses on microRNAs, which are involved in bone fragility in patients with diabetes. And my project is being funded by the Fidelio ITN, where we work with the help of funding from the Marie Curie Initiative. And the main focus of Fidelio itself in general is to understand why patients with diabetes are more prone to bone fractures. So it was a great experience for me at ECTS to share my work.
Definitely. But what did you present at the conference as such?
So over at the conference, I had a poster where we use a systematic review of literature approach in order to identify microRNAs that were found to be commonly expressed with respect to diabetes as well as bone. And when I say bone, we looked at the areas of bone relating to previous fractures or the occurrence of osteopenia or the occurrence of osteoporosis. And the reason why we went for a systematic review of literature approach was mainly because when I had done a literature review, there was very little information available in the whole field of science where we had identified microRNAs, or rather microRNAs were largely studied with respect to both diabetes and bone. So with the whole idea or the goal of having to develop microRNAs as biomarkers in order to better predict fractures in patients with diabetes, I set out with a systematic review.
Nice. But could you elaborate a little bit on what was the major implications of your findings then?
Yeah. So from the systematic review, we were very happy to identify 64 different mature microRNAs that were found to overlap between diabetes and bone. So this gave us a perspective of having to set these 64 microRNAs into, I like to call these 64 microRNAs a microRNA signature panel for diabetes and bone. And the next step would be to validate these different microRNAs in order to develop them as biomarkers for better fracture prediction in patients with diabetes.
So that could actually be used in the clinic in the future.
Definitely. And from the 64 itself, it's a lot more promising because we've identified about 6 microRNAs, which were found to be really strong candidates. And they also have promising roles with respect to glucose metabolism and bone remodeling.
That sounds really interesting, and also with a nice perspective for the patients as well. But what will be the next step in your career or your PhD? I know that you'll have some busy years ahead of you, but what have you planned so far?
Definitely, my primary goal would be to complete my PhD project and get my degree awarded. That's definitely number one on my mind, as well as I would like my findings of my 3 years of my PhD to be published in recognized journals. And I've always been interested in the field of basically anything to do with a bench to bedside approach has always attracted me. Yeah. So my primarily goal in terms of having to secure a job at the end of my PhD, or even to move in the field of research at the end of these 3 years of my PhD, would be to work closely related to diagnostic or treatment opportunities, where I would like to take something from the bench to the bedside.
That's also a really nice way of seeing the clinical science and the basic science, to see a match between that, because in my opinion that's also a part of the future way to think into science. So
it's always rather more motivating for me to see something being put into action instead of just basic science.
Yeah, exactly. I totally agree. And it has been really interesting to hear about your data and to hear about your future ideas, and I'm looking forward to follow your work. And I'm also gonna say thank you for taking your time to participate in this interview for HubLE. So thank you.
Thank you so much for giving me this opportunity. It was great talking to you.
You too.